started, good afternoon and good morning to those who are on. As we get started into this presentation that Mike had introduced, first thing we want to do is take a little poll to get an idea of what your concept of sterility assurance is. So, Mike, I believe you can open the poll. And the question is going to be, when developing a sterility assurance program, which of the below processes should be considered part of that program? Please select all that apply. Now, you should have the option within your control to select which of these various items: product and packaging design, material selection, manufacturing process design and supply chain, packaging, validation testing, and sterilization process and release testing. So we'll wait about 45 seconds, and then we'll move forward. Okay, so as you finish up your consideration on the poll here, I would like to pass the mic over to Martel Winters, who's going to cover some of the reasons why we're here today. And just to let you know that throughout the presentation, Martel and I will switch back and forth to cover our areas of expertise. Aha, there we go. Okay, so it looks like Martel, I'll go ahead and let you take it away for benefits. And as you can see here, many of you think almost every one of these elements is related to sterility assurance, and you are 100% correct. Martel?
Thank you, Aaron. Next slide, Mike. So some of the main points we're going to be focusing on today are here. So making sure that the finished product actually achieves the required sterility assurance level and that it's functional, and also an attempt to minimize project and product release delays. Hopefully to understand some of the inputs that affect sterility, and understand that the earlier in the design process that sterility is considered, or sterility assurance is considered, the better that process will be. Hopefully we'll gain some insight into the testing that's required for functionality, biocompatibility, and sterility, and then also the ability to educate others that you interact with regarding sterility assurance and how they can implement sterility assurance in the design and manufacturing teams. Next slide.
Really, guys, ultimately we're here to change a paradigm, and that paradigm is to get everyone to consider the fact that sterility assurance is much more than sterilization. And honestly, it seems like many of the folks that have joined this webinar today have a strong understanding of that based on the poll results. But there are many people within your companies that probably don't have that understanding. And to pick up on Martel's last point, one of the things that we'd like to encourage you all to do as you walk away from this seminar is understand the role you have to play in educating those who are around you. So if you look at the wheel that's presented here, ultimately, as noted at the start, this is a life cycle for sterility assurance, and it starts with product design and really product concept, and moves through a variety of topics, including material choice, packaging design, manufacturing process design, manufacturing environmental conditions, the sterilization process. Everybody knows that, and honestly, many people in probably the companies you work for, and certainly our customer base, understand sterilization as part of sterility assurance and little more. Then we have sterilization-related lab testing, and there's quite a few different places where that plays a role through the life cycle. And then sterilization, maintenance, and change control. So ultimately, the take-home point is sterility assurance, and the science of sterility assurance is much, much more than sterilization alone. Go ahead, Mike, to the next slide. So first off, we talked about product design. And really, if I think back to some of my own experiences when I wasn't working at Sterigenics, and right in the middle of my 20 years at Sterigenics, I spent several years away working for manufacturers, combination products primarily, but also medical device. And one of the key things was, and I was blessed to work at that company because they had an understanding of the fact that we had to start understanding the sterilization process the day we came up with the concept of the product itself. So the product has to be designed for sterilization, and some of the decisions that are really critical to make right away are sterility assurance level. What is your product going to be used for, and what level of sterility assurance do you need? Is the device or pharmaceutical product going to be single use? Generally speaking, a pharmaceutical product is single use, but perhaps the delivery device is going to have multiple uses and something you need to consider. Some might actually be re-sterilizable. And when we sterilize, is it going to be sterilized for clinical trials, or is it going to be sterilized, at the clinical site, or is it going to be sterilized in an industrial process? So you can choose to sterilize the product at the end use site, or you can choose to sterilize it either in-house or in an industrial contract sterilizer. Some of the common modes of sterilization that are at your disposal are moist or dry heat, thermal sterilization. You could look at vapor hydrogen peroxide. Radiation, a variety of different types of radiation can be used depending on what we're trying to sterilize. And gas processes such as ethylene oxide and nitrogen dioxide are also available. So there's a full array of different processes you need to consider really early in the process because it'll inform your decisions on the actual design of the product and delivery device. So we may be having to look at things like ensuring the sterilant has access to all the areas that are required to be sterile, keeping in mind that some things within the product, perhaps the drug itself, will go under a different sterilization process than the delivery device will be. So you have to be aware of where the sterilant has to have access and maybe where the sterilant shouldn't have access as we look at trying to sterilize, let's say, critical surfaces of a drug delivery device. We have to make sure in the product design that we've ventilated everything that does require sterilization properly so the sterilant can get there. Obviously, for gas and thermal processes, there may need to be some sort of ventilation for a radiation process. Perhaps you don't have to worry about that so much. And then the material factors need to be figured into product design. And honestly, more than anything else, materials and the product itself inform what choice you'll be ultimately having to take for the modality of sterilization you choose. If you move on to the next slide, Martel, we'll go into more detail.
So in selecting materials, we'll talk about this first. Clearly, the easiest thing to do is to base your selection of materials on literature or industry information. So what's available out there? There is a really good document that's published here in the US from AMI. It's called TIR 17. And the primary focus of that document is to provide sterilization compatibility with a wide number of polymers and a wide number of sterilization modalities. So it's a very good resource for an initial selection on materials that are going to be that you'd like to have sterilized through some form. The same thing with either active pharmaceutical ingredients or excipients, again, literature review, or you might have some internal data, again, regarding sterilization. And also either contract laboratories or internal laboratories. Contract laboratories particularly have a lot of experience with a lot of different types of products. And although they are very careful to not share secrets, generalities like this type of material seems to do well with radiation or does not seem to do well are things that they can often share. Now, sometimes it's not very straightforward. And if it's not very straightforward, then some feasibility testing is very useful. So in radiation, for example, too often when I hear of companies, they say, well, we tried radiation, it didn't work. And I say, well, what did you try? And I said, well, we tried 25 and 50 kilograms. And although 25 historically has been a very common minimum sterilization dose, there are a number of efforts going on in the industry, and I'll talk about a couple of these today that are helping to reduce that, to provide other options for much lower doses. So that's why I say here, somewhere in like 5 and 20, between 5 and 20 kilograde, to get an idea, at what point are you starting to see degradation of material? And also you can consider either ambient temperature or refrigerated temperatures. Under some, for some types of products and materials, refrigerated conditions or frozen conditions can make a significant difference on their compatibility with radiation. With moisture dry heat, we're consider and optimize cycle parameters. And again, we've had this typical approach to applying fairly long cycles for moisture dry heat, but really what is really needed, and I'll talk about that a little bit more later as well. And then consider non-traditional methods. There are quite a few out there, and at least consider and document. Now, I'm not recommending that you spend several years doing a research project with every sterilization type that you can find on the internet, but there is some wisdom in doing some initial screening through the modalities that are fairly well established. Next slide, Mike. So this is just one slide on the biological evaluation side, so the biocompatibility aspects. And the material characterization or material choice here has a lot to do with material characterization. And it's really important that as a manufacturer, you have really good relationships and understandings with your suppliers. And there are a number of things that are really important here. The manufacturing agreements, the focus there is largely on change control. The specifically pharmaceutical type products can be very prone to something that might be a simple change from a supplier standpoint, like location, maybe their supplier for the resin, or subtle changes in processing can make a significant difference in an extractable or leachable material. So having those agreements with really strict change control is critical. Composition disclosures are things that are nice to get from suppliers, but yet sometimes the composition of their materials might be proprietary. So if they're willing to share that, great. If not, then if they have any extractable and leachable material or data, then that is very helpful to you. And then also on the supplier, if they're supplying a component or a finished product, then also an understanding of processing aids and residual chemicals, a disclosure of those, or additional E&L data. Of course, safety data sheets can be useful. There are a few controls over how information is put into there, so sometimes they're not, but it's nice to have those. And also, sometimes your suppliers might have device master file information that they've submitted to regulatory bodies, and sometimes there is useful information in those. So these types of evaluations of suppliers up front, again, can really help to make sterility assurance an easier process and avoid delays later on. Next slide. Now you've noticed that Aaron and I have been talking a lot about sterilization, and so one question for pharmaceutical products, of course, is why are we talking about this? Why don't we just go with aseptic processing? And historically, aseptic processing has been a great go-to, and it still is. But we are seeing increased vigilance with regulatory bodies, where they are asking questions regarding how much did you really assess the ability to apply terminal sterilization to this product, rather than just assuming that that it won't work, or gathering no data and saying, well, aseptic processing is something I'm comfortable with, I just want to go forward with that. But there's some real advantages to terminal sterilization, and so there's no initial process simulation or media fill. There's no biannual process simulation required. There's no requirement for batch-to-batch sterility testing. In fact, I'm going to talk more about that later. Really no ultra strict microbiological manufacturing requirements, and you do have an actual sterility assurance level for the finished product. With aseptic processing, there is no SAL that can be applied, nor a contamination rate. There really is no output of a process simulation that gives a numerical value. The process simulation just tells you that the process is validated, but no output as a result of that. And so that as you look at these options for, should I do should I consider terminal sterilization, or should I go with aseptic processing? Really, it's part of a risk assessment. And as part of that, of course, you're leveraging, or you're comparing these options where is a change, there might be some change in the drug activity or purity, and if so, is that acceptable in exchange for terminal sterilization? And that's really what it comes down to, is functionality versus the real benefits of terminal sterilization. Next slide.
Okay, so moving on to a different topic. Once you've picked your materials and you've talked about product design, we also have to talk about packaging design. And really, packaging design, traditionally, everybody is aware that there is a package to help protect the product during transport, and also a package to help deliver the product during use in a reasonable order or expected order for use when we use the product. But we really ought to consider the sterilization as well. In fact, we have to, depending on the modalities we're using. It can become quite critical. So ultimately, the package has to suit the mode of sterilization. So for radiation, pretty simple. Most of the radiation we use will have no problem penetrating your package. And you can have a non-ventilated package, and it'll work fine. For some of the gas processes, such as EO or steam, you have to have the proper ventilated package. Ventilation, it's really critical, cannot be occluded by product or other packages in the sterilization unit container. So one of the classic examples of non-sterile situations I've seen over the years is where a company has attempted to perhaps save money on the ventilation side of their pouch and gone with a small ventilation patch instead of, say, the pictures that are here on this slide, which show an entire side of the package ventilated. And the thing was, they put those products into their shipper box and then stacked products one on top of another, and the polymer side of one package fit nicely completely over the ventilation patch of the next package, hence resulting in not only non-sterile product, but resulting in damage as well, because of the fact that the lack of ventilation caused the packages themselves to blow their seals up when a deep vacuum was pulled. So it's very critical to understand what sort of process your package will go under, and in fact, test for that as you develop your packaging. So the sterilant does have to have access to the areas that are required to be sterile. We have to ensure that the critical surfaces are exposed to the sterilant and properly ventilated. And the material obviously has to be compatible with the method. So there are a variety of methods out there that have material incompatibilities. And it's just important to be aware of that. There's no problem finding a solution usually to that material incompatibility, but you just have to be aware that some techniques can use certain types of packages and other ones can't. Packages may need to be able to withstand multiple processes. One of the choices that we have to consider early on is, are we going to allow for reprocessing or not? Now, in the pharmaceutical world, the vast majority of the time is going to be no, probably not. But there are some products that can undergo reprocessing. We just have to be aware of that if we're going to have multiple processes. And then finally, there have been some incredibly clever designs where the packaging itself participates in the sterilization. Give an idea, packaging could have vents in it that, or could have components within them that can absorb moisture. It can actually have components in it that deliver your drug to the product under deep vacuum. There's many really clever designs that have been developed over the years where the packaging actually plays a role in the sterilization and even the creation of the final device for pharmaceutical product, usually a combo device of some sort. Okay, we can go to the next slide. Marta?
So you have your proposed package design. Then some testing is good to do early on, and even if you're going into the full package validation, really whether you're doing aseptic or thermal sterilization, generally the packaging has to function in the same way once it's gone through whatever process that it needs. So generally the tests are the same that you're looking at doing after thermal sterilization. You may want to add an evaluation that if you, with some sterilization methods, sometimes there's a benefit in validating ahead of time that you can sterilize through 2 or 3 cycles and that the product and packaging are still fully functional. So that's something additional to consider. Determination of shelf life or stability testing, again, is largely the same. Now, one thing that we see regularly regarding package testing is performing sterility testing as part of stability or shelf life evaluation. And there's a really good FDA guidance document, and all 4 branches of FDA participated in this. So CDER, CBER, CDRH, and CVM all have their names on this document, and the title is Container Enclosure System Integrity Testing in lieu of sterility testing as a component of the stability protocol for sterile products. So in the introduction, just a clip of that says sterility tests are not recommended as a component of a stability program. So it's a short document, it's really good read, and it provides really good insight into really what is appropriate for your stability or shelf life data, and it has nothing to do with performing sterility testing and everything to do with verifying that the container or the package still maintains integrity. Once a product is sterile inside a container, it will always be sterile unless that container is breached somehow. Next slide.
Okay, so ultimately, once we've designed our package and the product and had the testing that we've needed to do to get us to the actual manufacturing process itself, we can start to really look at how do we design the manufacturing process for sterilization. We have to start with an understanding of the sterility assurance goal. I mentioned it at the very start of this presentation, but it's important to start thinking about the fact that your sterility assurance goal may actually be different for different components of the product. So you may have one sterility assurance goal for the drug product itself. And to Martel's point, that may be that we maintain drug sterility through an aseptic fill. But we may have a totally different and may be required to have a different sterility assurance goal for our drug delivery system, which is going to require some sort of terminal sterilization. So we have to understand what that sterility assurance goal is before we can develop the manufacturing process. And ultimately, like I say, product and packaging design must be finalized or very near finalized as well. We can go to the next slide. So the general goal of manufacturing process design is keep contamination to a minimum. It seems fairly obvious if we're going to try to sterilize product, but there are some considerations. While lower contamination of raw materials and a cleaner environment seemingly make things easier for sterilization, we have to keep in mind we're aiming here, assuming we're all running a business and not doing this just purely altruistically. We have to keep in mind that the product needs to be as clean as it is needed to be for the sterilization process chosen. Not as clean as possible, because as clean as possible, it doesn't really have an endpoint. We could really put a lot of time and investment into developing the most perfect manufacturing process that essentially has no contamination. While that might be needed, or as close as we can get to it for an aseptic fill, alternatively, if you're using the terminal sterilization method, there may be some latitude, depending on the product functionality when exposed to that method. There may be some latitude to allow for some microorganisms to be present. And we have to be aware of what our sterilization method can actually handle to determine how robust we need the manufacturing process to be from a microbial elimination standpoint. Some of the obvious things, handling, we do want to reduce handling as much as possible when humans are involved. Not only does it have the potential to contaminate the product, but it also introduces variability. As we all know, humans are not a piece of machinery that's just been cleaned. Humans have little quirks to them related to the fact that humans shed and humans have something coming out of their breath, and maybe the mask is put on perfectly, but maybe it's not, and so on and so forth. So reducing handling is helpful. And there's a couple other little points we want to point out here. One is be aware that there are some steps in the process, particularly if we're talking delivery systems, that may have bioburden reduction inherent in them and make it so prior to those steps you don't have to have quite as clean a process. And those might include extrusion processes, anodization processes, various cleaning steps, etc. But beware that if those processes themselves aren't controlled and validated, those processes may not be as robust as you think they are, and they may have variability that you're unaware of. So you do have to be aware that if there's any step you are actually planning to reduce bioburden with, you should develop some sort of testing to ensure the consistency of that process itself. Okay, move on to the next. All right, we're going to stop here, and if there's any questions, just take a quick moment. We can answer one or two questions from a design standpoint. You could type them in or send them to the not organizer, I guess it is an organizer. Mike, do we have any questions related to design?
So we're just going to give that 1 minute, Aaron, and see if we have any questions that come in. If not, we'll continue on with the presentation. There will be one more Q&A session at the very end of this presentation as well. So if you don't have a question right now, by all means, we'll be able to still get to some questions at the end. And
for sure.
So Aaron, right now, I'm not seeing any questions, so we'll just continue on. Great job explaining, guys.
All righty. We answered every question. That's amazing.
Excellent.
All right. Actually, Aaron, I do have one question, and I will direct this to either Aaron or to Martel.
Okay.
It says, in moist sterilization, if we declare that product is sterile, is sterilization validation, in a sterilization validation should be mentioned according to ISO, etc. So in moist sterilization, if we declare that a product is sterile, in sterilization validation should be mentioned according to ISO.
Yeah, in a general sense, if the sterilization process is going to be related to the actual delivery component of the system, ISO references are critical. That'll probably be judged as essentially part of a device, and you would be expected to follow the ISO standard for sterilizing the device. Alternatively, if we're using that sterilization method to sterilize the drug, it's important that you're aware of whatever the drug requirements are in the region that you are doing the sterilization in. So, for instance, in the EU region, there is an EMA guideline for sterilization of medicinal products, and you would also have to comply with that EMA guideline. Martel, any other thoughts from your standpoint?
No, you hit the one that I was going to bring up too, that there are regional requirements in addition to ISO that sometimes play a role. So those can be hard to track down sometimes, but they are present.
Okay.
Okay. So Aaron hit this very nicely, this idea of that sterility assurance really, when you're considering sterility assurance early on in the process like you should, that you can use those concepts to really drive the manufacturing environment. And Aaron hit on some of those topics on the previous slide. And big picture, the terminal sterilization allows for fewer controls and monitoring of the environment and of the product. Now, we're not saying that you can be lazy or sloppy. Sterilization is not intended to fix every problem that could happen. So it is still critical that there be good controls and monitoring systems in place. But certainly, if terminal sterilization is in place, then you certainly are permitted strategic reduction in these controls and monitors in the entire process to allow for the presence of microorganisms in a controlled fashion, just knowing that the terminal sterilization, the intent there, and the validation you perform will account for those. Next slide.
All right, so as we go into manufacturing environment, once we've designed our manufacturing environment, now we're really going to get into routine production. And I think this is something the average pharmaceutical company is highly aware of, so I'm not going to spend a lot of time on this, but we do need to monitor our manufacturing environment, and it does have a direct impact on sterilization of the product, or it certainly can if there's large spikes. So we really need to have levels set for what sort of contamination will allow from a microbial standpoint. Pre-sterilization, control charts are great to help detect trends and to detect unusual spikes. And usually over time, you will collect a lot of data and be able to use those control charts also just to detect drifts in the process. So ultimately, we should have alert levels and action levels. And thinking on action levels, these action levels largely should be planned on what you believe truly will affect sterilization. Alerts can tell us if a process is drifting, but action should be telling us, all right, we've got a real potential problem we have to address. When we do have out of specification events, we need to investigate them. And that investigation needs to be more than just counts. It's important for us to understand what types of organisms are showing up. I have a great experience, not great, actually, the opposite of great, but an experience I had many years ago where we had a surprise sterilization failure. And the fact was that they did have some unusual counts of organisms in the manufacturing area. The part that the facility, the manufacturing facility, did not look into was the fact that unusually, and for the first time, all of the organisms that showed up in that one batch of environmental monitoring were spore formers. And as you're maybe aware, spore formers can be quite challenging for many of our sterilization processes. So we had exactly the organism that the sterilization process really couldn't handle in large numbers. So critical to understand the type identification. And ultimately, based on that, not only counts and types, you need to determine what the risk to sterility is. And that risk will help drive your actions going forward, whether that's just a correction in the manufacturing process or if there truly is a risk to the product itself. Okay, move on. All right, so we've talked about a lot of things, and you can see that this is really pretty all-encompassing, and this sterilization and sterility assurance can encompass years of work from the very concept of the product up to the point where we're ready to manufacture the product. And due to that relative complexity and the fact that a lot of people don't think this way, and the fact that you need to be able to communicate the impact of sterilization to many different functions, from the engineers and project or product managers that are guiding the development of the product, through to the manufacturing engineers that are going to develop the manufacturing process, to the operations folks that actually have to execute that manufacturing process, and the quality engineers who will be checking the manufacturing process. All of these folks are not directly involved in sterilization, but it's critical they understand the sterility assurance impact they have. So one of the tools that I used to like to use, and I was blessed to be taught this from one of the companies I worked for, was the development of a sterilization strategy. And we developed this document really from the very start of product design and carried that sterilization strategy through to the very end and production of product. So this document can be years long and encompass all of the aspects that Martel and I have talked about. And when you're on a product design team and we're trying to bring a product to market, it's critical that we understand the timing, that we have the tests when we need to have the tests, that we know what potential roadblocks will be developed along the way and where our risk points are. So developing a formal sterilization strategy is a highly useful tool, and one I would encourage all of you to look into doing. And if anybody ever wants to talk about that offline, I'd be happy to help walk through some of the considerations for that document. I just found it invaluable as a communication tool. So ultimately, though, we have to decide, how are we going to process this product? And we need to walk through the various pieces. We go from manufacturing, and we have to talk about whether or not we're going to be sterilizing the product in-house or using a contract sterilization company. And if contract sterilization is used, we need to be aware of regulatory compliance, and we definitely need to have a quality agreement developed if there's any medicinal product involved. And ultimately, supply chain needs to be factored into the process design, the manufacturing process design. So we're talking about timing, the location, how that works with the supply chain. Is the location of the sterilizer somewhere reasonably located between manufacturing and distribution? Are you going to have air shipments? Is it going to be ground shipments? That impacts packaging. And these things need to be considered. And finally, consistency of process. Obviously, again, we all run a business also. And when supply chain people start talking about delivery, we need to understand what the commitment is with the sterilizer, whether it's done in-house or done with a contractor. Okay, we can move on. So ultimately, then we get into the pure traditional sterilization topics of process design and validation. And I have talked again about the more product and packaging, the more that the product and the packaging is designed for sterilization, the more efficient the process is likely to be. And obviously, the cleaner the manufacturing environment, also more efficient our sterilization process is likely to be. And I'm really not going to talk any more about this because we have several presentations on down the road today that will cover in much more detail sterilization process design and validation. These topics could cover days or weeks if we went deeply into them. All right, so move on to the next. Okay, Martel.
So like Aaron said, we're not going deep into sterilization validation, but there are a handful of things here that are either newer information that have been added to ISO standards or that are being available in the US from AMI. So I want to hit on some of these. So the sterility testing, the ISO sterility test or ISO sterility document, the 11737-2 was we did a new version of that last year, finished it up last year, and there are a couple of main points I want to pull out of that document. So the first is we added some guidance in there regarding method suitability or the BF test. That if it doesn't pass after multiple attempts, then performing a risk assessment and a rationale is acceptable to approve reduce inhibition, but maybe not to completely eliminating inhibition. We realize there are some products that are meant to be inhibitory in nature, and to require at the ISO level to require that acceptable data or perfect data be gathered in every case is just not realistic. And so we added some information regarding that. Also, we added a couple of bits of guidance here, and this goes back to the performing of sterility testing on fully sterilized product. So in the introduction, for example, we added this sentence here that performing a test of sterility on product that has been exposed to the complete sterilization process provides no scientifically usable data and is not recommended. And I'm going to talk about this a little bit more on the next slide, so I won't hit on that too much more right now. The bioburden standard, so 11737-1, previously there was the recommendation in the annex that 50% was a good value, but that was problematic, that it became a requirement, and so that's no longer in the standard. The recovery efficiency test for bioburden talks more about what really is important is more the consistency of the data and what is the intent for the data. Method suitability for bioburden testing is quite important as well as it is for sterility. So we now have a method suitability approach that we talk about in the bioburden standard, meaning a screen for any inhibitory substances in the bioburden test. And there's a brand new document that is not yet published. We just started working on it about a year ago on understanding and use of bioburden data. And that'll be called AMI TIR 106. And we're putting some good information in there on how to interpret data, setting alert and action levels, how to look at data in different ways depending on the situation, on what you're doing. So that'll be a good document to have out, and it won't be available probably for another year or so. Next slide. From a radiation standpoint, we are expanding the options for radiation for validation approaches. There's a new document that's almost out. It's AMI TIR 76, and for those of you that are familiar with radiation sterilization, the most common method by far right now for validation and maintenance of a radiation process is called VD max, for verification dose maximum. And this new version, there have been quite a few limitations right now on which sterilization doses you can select with VD max, and there have been no options for selecting different SALs. And TIR 76 will provide those opportunities for any sterility assurance level and for any sterilization dose. Then also AMI TIR 104, this is another document that we're working on that is not yet available but will be soon. So this is about transfer of sterilization between either different modalities, for example, from gamma to E-beam or vice versa, or from different facilities, for example, E-beam to E-beam, but different locations, and providing a lot of good guidance on being able to do that effectively. On the ethylene oxide side, just to make you aware, what we're seeing in the industry a lot in the last couple of years is a greater focus on more optimized sterilization cycles, rather than just defaulting to these long cycles because you can fit lots of product under that one family. Having more optimized cycle that are spending less time in the chamber and therefore having fewer residuals. So whether that's reducing the EO concentration, and there are a couple different ways to do that, or reducing the EO exposure time from going from the very traditional half cycle approach to either a cycle calculation approach or a BI bioburden approach. And implementation of either of those two options really can, not always, but can have a dramatic influence on the duration of the exposure time, and therefore can reduce residuals. And all of these topics are things that between Sterigenics and Nelson, we're very involved in the industry and working on these, and so would be happy to discuss these further if that would be of benefit. Next slide. So this is where I'm going to hammer on performing sterility testing, and I'm going to expand this a little bit, either as part of stability, part of your stability evaluation, or as part of release of product if it's a terminally sterilized product. So I've actually heard these. I was giving a presentation years ago in Canada, and I had just presented on why there are no scientific data behind performing sterility testing, either as part of stability or as part of a release criteria for a terminally sterilized product. And the fellow that got up just after me said, well, I still like to do it because it gives me a warm and fuzzy that things are good. Or it's how we do things, or more data are better. And sterility assurance is really, it's not about warm and fuzzy, it's not about historical significance or more data, but really it's about being smarter and how we approach sterility assurance, the overall sterility assurance of a finished product. The biggest issue with performing these tests at these times is, what do you do, besides the fact that there are no scientifically valid data you can get, because you cannot take data from these samples that were tested and apply them to others that were not tested. But what do you do if you happen to get a positive? So of course, the thought is, well, if you get a positive, that means that either the stability is, the packaging's bad, or the sterilization process isn't working, but that's rarely the case. If you are doing a lot of sterility testing as part of stability or as part of product release for a terminally sterilized product, eventually you're going to get a positive. And really you have to assume that it's because either stability or the sterilization process is failing, and it's virtually impossible as a laboratory to prove for sure that it's lab contamination. So you're left with this thing to have to deal with, and you'll be forced into taking action for something that really is not a problem. So that's my plea to the industry to think very carefully. Really, the purpose for sterility testing of a product is only in development and maintenance of a sterilization process, not as part of stability evaluation or as part of routine lot release. Next slide.
Okay, so we want to talk just briefly about, and I'm going to spend about 1 minute on this because I think we all know this, but ultimately you have to maintain a process, and we'll have a validated cycle specification. And as part of that maintenance each year or each period that you perhaps choose, we'll need to look back at the IQOQ to make sure that the qualification of our equipment is fine, and make sure that the performance over the previous year is fine, and ultimately revalidate based on risk in terms of the PQ. There are some cases where there's regulatory requirements for PQ revalidation. Perhaps in radiation you'll have a higher periodicity of reviewing the validation and re-executing it, but other standards may give you, and other modalities may give you different options in that regard. With regard to equipment, calibration, preventive maintenance, and demand maintenance, that type of thing is important as you analyze your process. And finally, with regard to change control, we need to have a process in place, know who's responsible, and execute appropriately. So we can move it on, I think, to Martel to wrap us up. Okay. Or Martel, was I going to cover this? I think you were, right?
Yeah, but really I'm just going to kind of summarize this. I'm going to summarize the summary and really just saying that what we're trying to really drive home here is that sterility assurance is something that is addressed from the early stages and continues on throughout the entire life cycle of a product. And the earlier on that you assess sterility assurance and use sterility assurance concepts to design a product and a process, the much better off you will be with regards to product timelines and actually the quality of the finished product. So I think we can just move on there. Mike, the last few minutes, if there are any questions that we want to answer.
Thank you, Martel and Aaron, and for the information that you presented today. We do have some questions that we have a few moments to ask. So I'll throw this first question out to you guys. When you talk about manufacturing trend analysis, can you mention some of the common types of metrics to trend?
Sure. Probably the most common types of metrics that people are trending for the trend analysis are related to microbial contamination. So doing microbial samples, often both of the air and critical surfaces. So there's a whole series of standards that nicely document the requirements in terms of what's allowed and what we should be checking from a microbial standpoint. And then similarly, from a particulate standpoint, and this gets more into just general control of the cleanliness of the air within the rooms as well as surfaces that you may measure. So these are primarily going to be the things that you check in the actual room or the areas where you're manufacturing. Now, the product itself, you can also be checking the bioburden on. And really, this can be part of your manufacturing process trend analysis. So we'll be checking the organisms that are actually on or in the product. And again, critical to not only track the trend of counts, but what type of organisms you're running across and whether those are within the expected norm for your manufacturing process.
And maybe I'll just add to that, what Aaron described is what is by far most common. The nice thing about when you're taking this overall sterility assurance program approach is that early on in the design phase, you might identify that you might have to do things in addition to those. I think what he's explained are things that always should be in place. But for example, if I might identify early on that this one component has a significant impact on the bioburden of the product or how the product is sterilized, then I might be implementing additional controls or monitors for that component that I'm doing on a routine basis as they're arriving or as where I'm assessing that component as a finished product. So that's the benefit of taking this approach, is there might be other things to consider.
Thank you, Martel and Aaron. Okay, we have a next question. What is the acceptable recovery efficiency if 50% is no longer in the standard? How often should recovery efficiency be performed?
Great question. So the answer to that question is that the problem we had with the 50% is that everyone was adopting that number without even thinking whether that value is appropriate for their situation or not. And let me give two quick examples. So if I have an EO sterilized product, and if I'm using some type of overkill approach there, then the bioburden of my product, there's little to no connection regarding the bioburden of my product and the extent of the sterilization process, just because of the significantly higher numbers that are in place that are on that BI that I'm using to evaluate or that I use for validation. So in those situations, then whether my recovery efficiency is 20% or 10%. If it's consistent, if the bioburden just does not come off very well, but if I have 10 data points and if they're all hovering around that 20% value, that's perfectly okay. And in addition to that, you might even ask the question, do I need a recovery efficiency? I just want to make sure my bioburden gives me a directional value. Is it 10 or is it 1,000 or is it 1,000,000? And the way the standard is written now, it allows you to make that assessment for yourself for what is really necessary for that product. And regarding repeating that, recovery efficiency is not a function so much of the people and things like that. It's more a function of the materials, the material surfaces, and the design of the product. So if the design changes, or something changes that might alter the surface characteristics of the product, then it's very wise to repeat recovery efficiency. But there are times where I think you could easily rationalize, and correctly so, that it's not necessary to repeat that. For example, if I have a new supplier of the same product and I have good characterization of the product, of a component, that component can change suppliers, for example, and if I have good characterization of that component, I would not see a need to repeat recovery efficiency.
Great, thank you, Martel. We're going to try to squeeze in one more question, and that question is, if you don't include sterility testing and stability assessment, what about shelf life verification?
So I'll take a shot first, and Aaron, see what you have to add there. So the purpose for stability usually is to show that or shelf life is to show that the packaging, whatever is supposed to maintain sterility, is still maintaining sterility, and also to determine that the product itself, the drug, for example, is still functional. And with regards to the component, the package or the container, if that package or container is still integral, if there are no leaks or damage to the packaging that occurs as you simulate aging or shipping or whatever, then the product inside is always still sterile. So you're looking at at physical test methods that give you good numbers regarding the strength or the barrier integrity of the package. Aaron, anything to add to that?
Yeah, I was really going to say exactly what you said, that there are quantitative physical tests we can do to prove the package itself is still functional, that are far better than a sterility test, which may or may not detect organisms that may or may not have wanted to try to come into the package sometime during the stability testing. The reality of it is that you'll get far better and far more useful data from the quantitative packaging, physical package tests and the test of the seals in the package than you would ever get from a sterility test.
Great. Thank you, Aaron and Martel. That is all the time that we have for today. Again, there was more great questions in there, and we apologize for not getting to them, but we can make those questions available to our presenters, and they will be able to get back to you when they have some time to get back to you and email those responses to you. So we hope that you will join us for our second webinar today at 1 p.m. Eastern time. That topic will be gamma radiation and nitrous dioxide sterilization, NEO, concepts and pharma applications. If you have not signed up, no worries. Just go to the Sterigenics.com events and go ahead and sign up now. We also invite you, as you see on the screen here, to take advantage of our other educational resources that we have to offer. Both Nelson Laboratories and Sterigenics both have white papers, articles, and on-demand webinars that you can always find on our websites. Again, we invite you to participate in our second session today, and then we also have 2 more sessions tomorrow on extractables and leachables and bacterial endotoxin. We thank you for joining us, and have a great day.